This presentation is mostly to recall what you already know, but some details must be added. So I'm not going to focus for most of the time on details you know. I will be just recalling them. And please treat this material as a sort of revision before we start real genetics. We already said that the mutation is a permanent change in DNA structure, but there are two main types of mutations. Point mutations, changing a sequence of nucleotides within DNA, and chromosomal mutations, which change structure or number of chromosomes. Due to the fact that you don't know enough about chromosomes yet, in this presentation we are going to focus mostly on point mutations, and the problem of chromosomal mutations will be discussed later. You know already that DNA base sequence encodes for RNA base sequence, and RNA in turn encodes for the sequence of amino acids in the protein. Sequence and composition of amino acids results in a particular protein structure, and the structure is what determines protein function, the role which the protein takes in the cell. What is done by the protein finally affects the trait we can observe in the organism. So this diagram shows how DNA sequence finally results in the presence of some particular trait in the organism. Point mutation is such a situation when the sequence of bases present in DNA is changed. There are many types of these mutations. We are going to focus on only one, when one base is substituted by another one. Such a mutation is called substitution mutation. Look at these examples. This is the original sequence of DNA in the gene. If the codon is TTC, it should be transcribed into the codon of IIIg in mRNA, and if it happens, what is a normal situation, then it encodes for the amino acid lysine in the protein. Lysine has its particular shape and functions. Now, what may happen if one of these bases gets changed? One of possibilities shown here is that one base is changed into something else, and in the result the codon in mRNA is also changed. So in this example, for instance, change of T in C in the second place of the codon results in change of A into G, and another amino acid is encoded. Instead of lysine, we've got arginine, which has different structure, so obviously this amino acid instead of this lysine will result in a changed shape of a protein and a different function. Similarly, if this letter in DNA would be changed in G, the codon will be changed in ACG, and the resulting amino acid threonine also has different shape than lysine and a different function. In both cases, doesn't matter. If the amino acid is changed into arginine or threonine, amino acids which result from the mutation are so different in structure and function that the final protein will be affected. Such a mutation is called missense mutation. However, remembering the name is not crucial. Understanding of the idea is what you should focus on. There are also other cases. You can see the example here. Let's say that the first base in this sequence of DNA is mutated into A. It results in UAG codon, which encodes for stop. The protein will be finished much earlier than it should. Such a short protein is non-functional, so actually the consequence is as if there is no protein produced at all. Such a mutation is called nonsense mutation. In both cases of missense and nonsense mutations, the result will be visible in the organism as a change of a trait. However, not always it happens this way. Sometimes the sequence of bases may be changed, but if you remember, some codons encode for exactly the same amino acid. Such an example is shown here. So, if the last letter from the codon, G, is mutated into A, the sequence of DNA is of course changed. But the resulting amino acid is exactly the same. This is lysine. In this case, mutation stays in DNA. The sequence of bases is affected. But there is no visible result in the protein. The protein is exactly the same, and the function of the organism is exactly the same. This is called silent or neutral mutation. It should give you the idea that not all mutations are harmful or in general visible in the organism. Some of them will be only visible if you look deeply into DNA sequence. Here is one of great examples to show you that quite often the change of a single letter in DNA may have a profound consequences. This is a case of sickle cell anemia. However, the name is similar to the name of conditions when you get an iron deficiency or something. This is not directly related to that because the background for this disease is genetic. There is one mutation in, please attention, this is what you have to remember, in a gene encoding for hemoglobin chain. I hope that you know hemoglobin. This is the protein which is responsible for carrying oxygen within red blood cells. You can see how the normal red blood cell should look like. Inside it should be packed with hemoglobin molecules carrying oxygen to body tissues. Such a normal hemoglobin is encoded by the gene which has in one place such a sequence, CTC, encoding for the glutamic acid, amino acid present in one place in the whole protein. The mutation we are talking about changes only one letter within this sequence. T is converted into A. If such a mutation hits DNA, then in consequence RNA sequence is also changed. Instead of GAG, you get GUG codon, and while the first one, the correct one, encodes for glutamic acid, the incorrect one, the mutated codon, encodes for valine. This amino acid has different properties, and the resulting hemoglobin will have different structure and will function differently. What happens to the protein? The first thing is that such a mutated type of hemoglobin will not bind oxygen well. So in consequence, the organism will be not supplied with sufficient amount of oxygen. It means that the person suffering from sickle cell anemia will live in a constant state of oxygen deficiency. Of course, it will affect the most tissues which have high oxygen demand because they perform high level of cell respiration, the nervous tissue and muscles, especially during exercise. So such a person will be weaker, will have problems with performing stronger exercises, and in general, development of the whole organism will be also impaired. But this is not the only problem with this protein, not only that it carries less oxygen. The second problem with a mutated protein, the consequence of only one amino acid change, that instead of staying in the normal state, molecules of mutated hemoglobin will join, forming long abnormal chains. And within the red blood cell, these chains will result in abnormal shape of a red blood cell. Can you see them? The shape of them, sickle means sharp. The shape of them is not only relatively easy to recognize, it results in the fact that these abnormal red blood cells tend to clump much easier than normal ones. If they clump, they block blood flow, especially in smaller blood vessels. Many tissues in the body will be not supplied in oxygen at all. It results in strokes, pain, and abnormal development of the body. So overall, combination of these two consequences of a changed protein structure results in a deadly disease which usually leads to death before 20th year of life. So you can see, and you should be able to explain on this example, how mutation of a single base in the gene can result in such a huge effect in a person's physiology. It was also discussed already during the class. It was said that not all mutations will have the same effect. We said already that in some cases, change of DNA sequence will not result in the change of codon meaning. So the codon may have different bases, but it may still encode for the same amino acid. And if it happens like that, then amino acid sequence remains the same, protein remains the same in its structure and function. So finally, the trait is not changed. This is what we call silent mutation. If codon is finally changed, then we also may have diverse effects. Sometimes stop codon will be produced. In this case, actually, protein is not produced at all, so obviously it will change the physiology of the organism. How deep is effect, however, depends on which protein was removed this way. If amino acid sequence is changed this way that protein is still produced, it may have its structure changed a bit or strongly. In case of structure changed only a bit, like one amino acid is replaced by another one which is only slightly different, then protein still may have more or less the same function, but for instance a bit weaker. So the trait in this case is of course changed, but it may be not a huge abnormal effect for the organism. On the contrary to that, if protein structure is strongly changed, it actually may result in one of three well-visible consequences. Protein function may be impaired, and this is what we said, for instance, about sickle cell anemia. So actually, the abnormal hemoglobin still works, but not fully, and it results in a changed physiology, much less oxygen present. Sometimes a protein will gain new function. Actually, you can also say that mutated hemoglobin in sickle cell anemia is a similar example, because this mutated protein not only will carry oxygen not well, but also will lead to clumping of red blood cells. So it is also another change in the trait, also quite bad for the organism. And sometimes a mutation leads to the total loss of protein function. It happens, for instance, in so-called cystic fibrosis, the disease which in Polish is called mukowiscydoza. In this case, the chloride channel is not functioning, so chloride ions are not transported in cells producing mucus. And in all tissues and organs, when mucus is produced, abnormal type of mucus is produced, and the whole organism is affected by that, by the total loss of the channel function. Overall, we may say that change of DNA base may have a huge diversity of consequences. It depends on the type of a change and the type of a protein it affected. What causes mutations? This is something new. This was not discussed yet, so please try to focus more from this moment. The general reason for mutations appearing is the way in which DNA is replicated. If you remember this process more or less, it was stated that when one DNA strand is exposed during DNA replication, then new nucleotides will arrange along this strand according to complementary base pairing, and then enzyme, DNA polymerase, will glue these new nucleotides together to form the complementary strand. It normally happens like that, but the DNA polymerase will not know which nucleotides are here. So it sometimes happens that one of these nucleotides is not the correct one. Just by the accident, something else appeared here. And if it happens like that, DNA polymerase will not know that the nucleotide is incorrect and will incorporate it into the new strand. It happens each time DNA is replicated. These mistakes are not very frequent, but taking into account how long is DNA in each cell, overall, replication of DNA material from a cell always results in some mutations. Some of them will be found and corrected by proofreading of the cell, but not all of them. And this is the crucial cause for mutations appearing in DNA. But there is another group of factors which can lead to formation of mutations. They are called mutagens, various external factors which can knock out some bases out of DNA or change their structure in such way that later during replication the DNA sequence will be changed. To this group, some chemical substances belong. Ethanol is one of mutagens. Many toxins are mutagens. Substances present in, for instance, nicotine smoke are mutagens. Their presence increases probability of such changes in DNA strand that finally it will result in a change sequence. Another category of these factors is irradiation. Different types of irradiation may result in a change in DNA structure. Some of them will change the structure of a base. Some of them will destroy the base thoroughly. So later, when DNA is replicated, the DNA polymerase will incorporate random base in this place because it will not know what should be present there. And finally, there is a huge group of viruses causing mutations like herpes virus, responsible for development of cervix cancer. Viruses incorporate their own DNA into the genome of a host, and by doing that, quite often they change a sequence of bases in such a way that may lead to mutations being preserved in DNA. So what does it mean? If a mutation happens for the whole organism, what can be the health consequence for the organism affected by a mutation? The first thing to stress is that mutations are random. They, in general, occur without no particular rule. They may happen in the part of genome which encodes for no gene, but they can also hit real genes. It cannot be predicted where some mutation is going to appear. If a mutation happens in the part of genome which encodes for something crucial, it may happen that the change of a cell functioning is so strong that the cell will simply die. It may die due to its abnormality and being not able to work, but sometimes if mutation is detected by protective mechanisms, the cell will be forced to die by so-called apoptosis. This is sort of suicide of a cell controlled by organism. In other terms, organism has ways of detecting abnormal cells, and many mutations will finally lead to the fact that organism will send signals to abnormal cells to die. If mutation happens in one of genes which are called proto-oncogenes, the genes controlling cell divisions, it may lead to formation of cancer. If mutation affects genes which normally should control how frequently and at which moments the cell is dividing, then it may happen that this control, which should be normally present, is lost. Such a mutated proto-oncogene turns into oncogene and finally may lead to the formation of cancer. It may happen in almost any place of the organism. So there is no specific cell in which such a mutation must be present to result in a cancer. It may happen everywhere. On the contrary to that, if some mutation happens in the reproductive cell, in a gamete, it may be passed to the offspring. And if it affects some crucial gene, it may lead to the presence of genetic diseases. I would like to stress, and this is very important, that to result in genetic disease which is passed to the offspring, the mutation must be present in the reproductive cell. In other terms, the person will get genetic disease only if it is inherited from parents, then it will be present in all cells of the organism, or if by some accident a sequence of DNA in a gamete was mutated, and then this gamete provides the abnormal gene to the developing new organism. Otherwise, if mutation happens in one of many cells of a whole organism, it may affect functioning of this organism. It may lead to abnormalities in functioning, but will be not passed to the offspring. So these two types of mutations happening anywhere will not result in passing the trait to the offspring. But mutation in the gamete may result in genetic disease, which will be later passed to next generations. There is finally the fourth possibility. If mutation hits a gamete or developing embryo or fetus, it may kill some cell which is crucial for further development of the organism. In this case, such a mutation may result in abnormality of fetal development, and the fetus formed may be abnormal. It may lack some body parts, or they may be misshaped. Some additional structures may be present. In general, mutation in developing embryo is much more dangerous for the whole organism than it is in the adult one. Because in adult one, there are many cells, and they usually can be easily replaced by others. But if a mutation happens in a very small developing organism, when there are only some cells present, destruction of one of them may finally affect the whole future development. But actually, we cannot say that mutations are overly harmful. On the contrary to that, they are really needed from an evolutionary point of view because they cause variation. They form so-called new alleles, new forms of genes. Without mutations, the gene present would always stay the same, and due to mutations, almost unlimited variety of them may be formed. So, of course, mutations can be harmful, but sometimes they can also be beneficial, forming new traits, new skills, new structures. Or, of course, sometimes it can be both. And for instance, the sickle cell anemia, which was already mentioned, is generally harmful in many cases. But in some particular situations, presence of this mutation is beneficial because in organisms with this mutation present, malaria will not develop. So in malaric regions, this mutation is relatively common because it protects from malaria. So it cannot be stated clearly that mutations are bad or good, but we can be sure that if there are no mutations, there would be no variation among organisms and no evolution would happen. Because, as you probably know, evolution is choosing organisms which have better adaptations. Having identical adaptations, all organisms would be exactly the same. What we said previously is absolutely everything what you should know at the moment about mutations in DNA sequence. Here I only want to mention a bit about chromosomal mutations. But this is something what will be not discussed right now but it will be incorporated into the next presentation, which will explain clearly what are chromosomes, what's their function, and how they work. Then you can understand how abnormalities in them are important. So in general, chromosomes are pieces of DNA present in the organism, large pieces of DNA. And their mutations, on the contrary to the previous category, their mutations are big changes, duplicating or removing huge parts of DNA in the cell, thus resulting in much profound changes to the whole organism. What are exactly categories of chromosomal mutations and what are consequences will be explained in the next presentation. Thank you. In the homework, which will be created based on this presentation, you will also find information which pages from the textbook you should read and which exercises should be done to check if you understand everything. If you've got any problems, questions, things you would like to discuss with me, please write them down listening to this presentation and I will find a moment to discuss it with you somehow.